Students, today we shall be talking about the structure of gills, particularly in the case of fishes. As we all know that the gills are the respiratory organs in fish and other aquatic organisms. The gills are the main site of the exchange of gases in almost all fishes, and there are 5 to 7 pairs of gills in the case of cartilaginous fishes, whereas 4 pairs in the case of bony fishes, which never exceed 5 pairs. Gills, when we define gills, we define it as the filamentous, feather-like organs, richly supplied by blood capillaries for the exchange of gases, that is oxygen and carbon dioxide. These lie within the gill clefts and appear as vascular, filamentous lamellae-like outgrowths from the anterior and posterior walls of the gill clefts or gill slits. The use of the gills is secondarily adapted from the filter feeding and the current generated by the cilia in primitive chordates and agnathans. Awareness of the gill respiration nearly dates back about 4 centuries. When we talk about the classification, the Linnean classification, which distinguishes fishes from other vertebrates by the use of gills and respiratory organs. Otherwise, before that, all the organisms living in water were described as one group, whereas the Linnaeus, on the basis of the gills, he classified the fishes as different vertebrates from the others. Let us have a little bit idea. I use the term gill cleft or gill slits. Then we have to know about what spiracle means. What are gill arch? What are gill rakers? What are gill filaments or gill lamellae, which are primary as well as secondary? So I shall be introducing you, before going on to the topic, I shall introduce you that what these all terms means. Now, looking at this very diagram, we are seeing this is a cartilaginous fish, and herein we are seeing these pores, 1, 2, 3, and 2 pores are missing, just hiding behind this very fin, this pectoral fin. So here in this case, we are seeing that this is a cartilaginous fish and it has 5 pairs of gill slits. Now, these are the slits which represent that behind it the gills are present. But in this very case, in the case of bony fishes, what we are seeing here, there is only one slit is only one, but inside it, this is called as the opercular bone. Now this is only one pore externally, but inside it every gill has a slit, which are up to the 4 pairs. Here in this case, these are 5 pairs, and in this case these are 4 pairs, but externally it is visible only one. But here in this very case, the cartilaginous fishes, we all know that these are the fishes which have cartilage as an endoskeleton, whereas when we talk about the teleost, these have we call them as the bony fishes, and these have the bony endoskeleton. One thing is more worth to mention here, that in the case of these very sharks, these are the slits here, and there is one spiracle also. It is just behind the eye, and it is a pore just behind the eye, which is called as the spiracle. But in this case, only the opercular bone is present externally, but no spiracle is present in the case of bony fishes. The gill clefts or the gill slits on each side open internally into pharynx. These open internally into pharynx by wide internal branchial apertures, and outside by narrow external branchial apertures. In the case of sharks, the gill slits are laterally situated, while they are ventrally placed in case of the some other cartilaginous fishes. In the case of sharks, they are laterally placed, whereas in the case of flat fishes, they are ventrally placed. The gill slits in the case of bony fishes, they are covered by operculum, while the operculum is absent in the case of cartilaginous fishes. There is no operculum in the case of cartilaginous fishes, whereas in the case of bony fishes, operculum is present, which is called as the gill cover. So the gills are covered by a bone called as the operculum. So this is the operculum. Herein it is demarcated like this. This is the operculum, the bone. Now, um, now this is a diagram which clearly shows the gills inside the gill, this very gill cover or operculum. Now we are seeing here in this very fish, this is the first gill, this is the first gill, this is the second gill, this is the third gill, this is the fourth gill, this is the fifth gill. So in the case of bony fishes, the gills never exceed 5, but mostly they are 4 in pairs. And in some cases, the first gill is very much reduced in size, and it looks like this. It is very much reduced in size, and this is said to be as pseudobranch. Now, branch, we use the term for the gills, whereas when the gill is reduced very much and it loses the function of the respiration, then it is a pseudobranch. Though it has some other functions in the embryonic stage, it has the function, but in the adult stage, it loses its function of the respiration. Earlier, I showed you the gills inside the fish, and now herein, this is the gill. This is the gill of Sirhinus murigala. Herein, now you have to first of all learn and to know about the structure of the gill. What we are seeing here in this very case, we are seeing here, this is the gill arch. Now this very arch, one we call it as a circle, incomplete circle is called as the arch. Now this very arch is not so circular, but the gill, the bone which is almost of the shape of a semicircle, it is called as the arch. So this very bone which is semicircular in shape, it is the gill arch. And inside this, these very teeth-like structures, they are called as the gill rakers. But students, keep in mind, that these very gill rakers, they differ from species to species. Now herein, what we are seeing here in the case of Sirhinus mrigala, which is an omnivore fish, herein we are seeing short and stumpy, you can say, gill rakers. But in the case of Hilsa elisha, which is a planktivore, herein what we are seeing the filamentous gill rakers. So these very gill rakers also help in the feeding of the fish, because these very gill rakers help to filter the plankton. That is why they are filamentous in this very case. But in this very case, they are short, they are stumpy, or in the carnivores, they are strong, teeth-like, and they help in the feeding of the fish. So now the question arises, then what is the function of the gill raker in their respiration? Yes, this is an important question, that why the gill rakers, we do study the gill rakers in the case of in the case of this structure of the gills. Now they are the part of the gills and they protect the gill filaments. Now actually, what are the actual site of the respiration? They are the gill filament. Now this very side, these are the gill lamellae or gill filaments. We call them as the gill lamellae or gill filaments. They are primary gill filaments and they are secondary gill filaments. So what we learned from here, that the first of all there is an arch. On the inner side of the arch, gill rakers are present. On the outer side of the arch, gill filaments are present. Gill rakers differ from species to species depending upon the feeding habit of the species. And secondly, the gill rakers prevent these very some particles which are very much injurious to the gill filament. They protect these very particles from entering towards the side of the gill filament, so that they protect the gill filaments. So gill arch, gill rakers, and then on this very side, minute vascularized filamentous, you can see, structures, we call them as the gill lamellae or gill filaments. Now in this very case, you are seeing in the case gill rakers, in the case of Hilsa elisha, they are also long and thin, and the filaments, though they are also long and thin. But herein, what we are seeing now here, we are seeing the gill, a complete gill. For example, I will take this very portion I will take out and zoom it. It will look like this. It will look like this. On this very side, the gill rakers are present, and now the gill filaments are like this. For example, this is a gill arch, and these are the gill rakers here, and these are the gill filaments here. But you should keep in mind that the gill filaments are almost v-shaped. One gill filament, it is approximately like this. So I am taking this very and showing you here what we are seeing now. We are seeing now that on the inner side the gill rakers are present, and two gill lamellae, and two gill lamellae are attached to. So one gill comprises of two gill lamellae and gill rakers. And now this is the arch. This is the arch here. Now here we are seeing it here in a, on a bigger, you can say, diagram. Here what we are seeing here, the gill rakers, then this is the gill arch, and then what we are seeing, this is one gill lamella, this is the second gill lamella, but this is called as this one is whole, as a whole it is called as holobranch. This is called as holobranch. And when I study only half, I'm studying only half. For example, this one, I'm studying this one only. This becomes the hemibranch. This is hemibranch. So the whole gill is called as holobranch, and half of the gill is called as the hemibranch. So a gill comprises of the primary gill lamella and secondary gill lamella. Now secondary gill lamella are present on the primary gill lamella, so as to increase the surface area, that we shall be studying a little bit ahead. So we got to know about the gill structure. The firstly, it is the opercular bone in the case of bony fishes. Underneath, the gills are present. When we take one gill out, it is comprised of a gill arch, then gill rakers, then gill filaments, which are primary gill lamellae and secondary gill lamellae. And the gill rakers vary according to the food and feeding habit of the fish, whereas in the case of cartilaginous fishes, there are only gill slits, no operculum is present. Now this is the complete diagrammatic, you can say, representation of the gill lamella. Herein, this is the gill raker, and this is the afferent branchial vessel. These are the secondary afferent vessels. Now this is one thing, this is the interbranchial septum, that is the space between two lamellae. The space between the two lamellae, it is the interbranchial septum, which I am coloring it as the red. This is interbranchial septum. Now students, this also varies according to the food and feeding habit as well as the behavior of the fishes, whether the fish are actively swimming, fish belong to the hilly regions, warm water or cold water fishes. This very septum is can be shortest, or this very septum can elongate up to the half of the gill lamellae. Now, secondly, um, a pair of spiracle is present. This I was talking about this spiracle. The pair of spiracle is present in elasmobranchi anterior to the first gill. Now, for example, the gill slits are present here in this very case. For example, five gill slits are present. So this is present anterior to the first gill slits, and it corresponds to vestigial first gill slit. So this is a vestigial gill slit which has become vestigial, and now it is spiracle in the case of contracthes. Spiracle is absent in the case of actinopterygii, and it is replaced by, I was talking about that, it is replaced by the pseudobranch, which is free in some fishes but skin covered in the case of others, which can be free or skin covered in some fishes. Now talking about the gill slits. The lateral wall of the pharynx are perforated by means of series of slit-like apertures, the first of which is called spiracle and lying between mandibular and hyoid arches. So in the case of contracthes, spiracle, we are talking about the contracthes. So this is the mandibular arch, and this is the hyoid arch. So in between these two, between the first spiracle lies between the mandibular and the hyoid arch. So in this very position, the spiracle is present, which is depicted externally or morphologically. The second eye or the hyoid cleft lies between hyoid arch and the first branchial arch. So second or the hyoid cleft, we call it as the gill cleft or the hyoid cleft. The second gill cleft is called as the hyoid gill cleft, which lies between the hyoid arch and the first branchial arch. So this is the first branchial arch, and this is the gill lamellae. This is the gill lamellae, first gill lamellae. So between hyoid arch and the first branchial arch, the first gill slit is present, while rest of the gills are situated between the succeeding branchial arches. So rest of the gill slits are present in the respective branchial arches. Now talking about that, how many gill clefts are the gill slits are present in the case of fishes. So it is just for the information, for your information, that in some cyclostomes such as Delostoma, there are 14 pairs of gill slits. In the case of Petromyzon, for example, if this picture is shown here, in the case of Petromyzon, 1, 2, 3, 4, 5, 6, and 7 pairs of the gill slits are found. In the case of Myxine, it is 6 pairs. In the case of cartilaginous fishes, 1, 2, 3, 4, 5. It is 5 in the case of cartilaginous fishes. But two exceptions are there in the case of cartilaginous fishes. The Hexancus, there is a species called as the Hexancus, which has 6 pairs of gill slits, and Heptancus has 7 pairs of gill slits. This is the Hexancus having the 6 pairs of the gill slits. And in the case of bony fishes, they can never be more than 5, and mostly they are 4 pair. When we talk about the sharks and rays, they typically have the 5 pairs of the gill slits that open directly to the outside of the body. Though some more primitive sharks have 6 pairs, and the broad nose, they have the 7 gill slits, and it is the only cartilaginous fish exceeding this very number. Now, talking about the pseudobranch, I talked about this very pseudobranch, that it is the first, you can say, gill arch which is reduced. So when we talk about the pseudobranch, it is also called as a pseudobranchia or pseudobranch. It is reduced. First gill arch. This one. This we call it as the reduced first gill arch. This is the first gill arch which is reduced. In some species, the pseudobranch with the hemibranch structure is located inside the operculum. But in the case of eel, pseudobranch is not present, and also in the case of some catfishes, the pseudobranch is missing. So pseudobranch is not present in the case of eel and some catfishes belonging to family Siluridae, and the featherbacks belonging to family Notopteridae, belong to Notopteridae and Siluridae. When we talk about the pseudobranch, in many actinoptergians such as Catla, Hadean pseudobranch consists of a series of gill filaments which are present anterior to the first gill slit. So in the case of Catla, the gill filaments are present anterior to the first gill slits on the pseudobranch. It may be covered or with the layer of the, it can be free or it can be covered by the layer of the mucous membrane. And pseudobranch dwells during the early embryonic stage. It dwells during the early embryonic stage and has respiratory function. It has respiratory function in the early embryonic stage, but respiratory function, I have told about this in the earlier slide, that this very respiratory function is lost during the adult stage. Pseudobranch is absent in species like Velagonia atu, as I told you, in some catfishes or featherbacks, Velagonia atu, Mistus or Notopterus or Notopterus chitala, Notopterus, you can say species, and Channa species. It is said to be absent in those very fishes in which chloride gland of the eye is not present. It is also related to the chloride gland of the eye in those very fishes wherein the chloride gland is not present. The pseudobranch is also absent in those very fishes. So it is said to be useful in filling the gas bladder and regulation of interocular pressure. Interocular pressure. It is useful in filling the swim bladder, which is used to maintain the buoyancy in the case of bony fishes, and regulation of the interocular pressure, and that is what we call it as the eye, the pressure, the eye pressure. Histologically, pseudobranch contains large number of acidophilic secretory cells, which probably produce carbonic anhydrase enzyme, which particularly produce the carbonic anhydrase enzyme. So they are also known to contain the acidophilic secretory cells known to produce the carbonic anhydrase. And pseudobranch also receives oxygenated blood directly from the dorsal aorta, and has vascular connection with the internal carotid artery. So oxygenated blood is received from the directly from the dorsal aorta with and having the connection with the internal carotid artery, and it may also serve to increase the oxygen concentration in the blood. It also increases the oxygen concentration in blood, which goes to the brain and eye through the internal carotid artery. So in order to maintain the oxygen concentration in the blood, in the which is going toward the you can say brain and the eye, the oxygen concentration is maintained with the help of the pseudobranch. So this is the important point to note. So this is just a fish which is depicting that okay, this is the operculum, and this is again the gill of the fish with the gill rakers. Okay. Now we shall be discussing a little bit about the complete gill, which is called as holobranch. Holobranch. Incomplete gill, that is half of the gill, it is hemibranch. Okay, so the gill which has lost, which has become vestigial, it is pseudobranch. You have to keep in mind all these very terms. Pseudobranch, the gill, the first gill, which is vestigial now, which has lost its function of the respiration in the case of adult, but in the case of you can say early stage, embryonic stage, it has the function respiration, but it has lost the respiration function of the respiration in the adult stage. That is pseudobranch. The complete gill, holobranch, half gill, hemibranch. Okay. The complete gill are this is the complete gill. This is the complete gill. The holobranch consists of the gill arch. This is the gill arch. The complete gill consists of the gill arch, which is supported by the cartilage or the bone. So a cartilage or the bone supports the gill arch. The gill arch bears gill rakers. So these are the gill rakers which are in the case of the which are supported by the gill arch. Toward the inner side, they are toward the inner side, and gill filaments. These are the gill lamellae or gill filaments toward the outer side. So these are the primary gill lamella, and on the primary gill lamella are present the secondary gill lamella. But this is the primary gill lamella overall, and this is the secondary gill lamella. So half of the gill, it is the hemibranch. Each row of these filaments form the hemibranch or half gill. So gill raker toward the inner side, and gill filaments are the gill lamellae toward the outer side. When we talk about the structure of the, we shall be particularly talking about the structure of the Teleostean gill. So, Italian scientist Marsilio Malfigi in 1628 to 1694 made comparative observations of the gill structure, and in 1665 Samuel Collins described the importance of the oral valves to the respiration in the case of flatfish. In addition, being the principal aquatic respiratory organ of the fishes, gills are also primary site of osmoregulation. We cannot deny the fact that they are only the sites of the respiration, but they are the primary sites of the osmoregulation and nitrogen excretion as well, and have the major involvement in the acid base balance and hormone metabolism. So the four functions are the four most important functions. The number one is respiration, number two is osmoregulation, number three is acid base balance in the case of fish, and then hormone metabolism. These four things are associated with the gills. Now we do have the salt concentration, maintenance of the salt concentration in the case of the marine fishes. That is also one of the important, you can say, features of the gill, but we shall not be discussing at length. We shall be directly discussing on the structure of the Teleostean gill. We have discussed this very image in our in the start of our slide. The fish, this is the operculum. Inside the operculum are present the gills, separated by the gill slits, or the gill slits. This is the gill, these are the gill rakers, and then this is the TS of the gill, which we have discussed. Now, talking about the structure of the Teleostean gill, a complete gill is known as the holobranch. It consists of bony or cartilaginous arches. The anterior and the posterior part of each gill arch possesses flat plate-like filaments. Plate-like filaments. Each holobranch consists of anterior, that is oral, and posterior, that is aboral hemibranch. So it consists of the complete gill consists of the, you can say, a complete holobranch consists of the hemibranchs, and uh, it also possesses the gill rakers and gill filaments. When we talk about this the structure of gill has been studied extensively in the case of Indian fishes by light transmission and scanning electron microscopy, and the gill comprises of the gill arch, gill rakers, gill filaments. So what the gill comprises? It comprises of the gill arch. Number one, it comprises of gill arch. We shall be discussing one by one. Gill rakers, then gill filaments. The gill filament, these are the three important things which make a gill. So the gill, when we talk about the gill filaments, they are the primary gill filaments and they are the secondary gill filaments. First of all, we shall be talking about the gill arch. Now, as we are seeing here, that gill arch is one of the important unit which bears primary and secondary gill lamellae. This is the gill arch. This is the gill arch. It bears the primary and secondary gill lamellae. Now, actually, these very filaments are actually in this V shape, on like this, on the arch, like this, like this. So on the inner side of the gill arch, the gill rakers are present, and on the outer side, the gill filaments are present, which we called as the gill lamellae as well. Now, when we see the internal structure of the gill arch, we see that it is composed of the pharyngobranchial, then this is the epibranchial, and this is called as a ceratobranchial, and then a small bone called as the hypobranchial, and a small median unpaired basibranchial. Whereas other bones are in pair, and this very basibranchial is unpaired in the case of this very gill. When we talk this very the epibranchial and the ceratobranchial elements of each branchial arch bears two rows of gill filaments of the two hemibranchs of a holobranch. So when we talk about this very thing, which is comprising this is in Gill Raker, and then this is the Gill Arch, and then this V-shaped gill filament. This is a complete holobranch. And when we talk about this very one filament only, this is a hemibranch. Which are the seat of the gaseous exchange? Actually, these are the filaments which are the seat of the gaseous exchange, and it encloses afferent and efferent branchial arteries and the veins. So in the case of teleost, this is actually but what we are going to see in detail. This is actually the we have discussed this in the earlier slide as well, but herein we will discuss it again, that this is the gill arch, which can be bony or cartilaginous. This is the gill arch. Here on this gill arch, on the inner side, the gill raker is present, and two gill filaments, which are called as a primary gill filaments. But on these very primary gill filaments, these very odd growths on the upper as well as the lower side are present. We call them as a secondary gill lamella. Now we are not going to discuss it here. We will discuss it in the next part, that is the primary and secondary gill lamellae. So these are the primary efferent vessel is also present, then the efferent branchial vessel is present here, and then the secondary efferent vessel is present here. So I mean to say that it is very much vascularized, and actually it is the seat of the gaseous exchange. The gill arch does not play any role in respiration. The gill arch is important unit and bears primary and secondary gill lamellae, which are actually the seats of the respiration. The gill arch is only a supportive in function, which supports the gill rakers as well as the gill filaments. Now, the gill raker, we have talked about this very gill raker, that the inner side of the gill arch are present the gill rakers, which are short and stumpy structure in the case of bony, supported by the bony elements. In the case of Serranus marigula, they are short, pointed, whereas in the case of Hilsa alicia, they are long filamentous, because this is a planktivore fish and this is the benthic omnivore fish. Gill arch projects across the pharyngeal opening, so it is present across the pharyngeal opening, and these are modified according in relation to the food and feeding habit. For example, this is the here in this very case, this is the gill of the common carp. This is the gill of the Schizophorex isosceles. Here we are just to mention here, that to show that these are the gill filaments and these are the gill rakers, because these vary according to the food and feeding habit of the fish. The mucous cells of the epithelium help to remove sediments from the covering epithelium in order to enable the taste buds to function effectively and to sense the chemical nature of the food passing through the gill sieve. So it acts as a sieve for the food material. Actually, the food material, they get entangled in the gill rakers. They are sent toward the alimentary canal, whereas the water after filtration, they are shown the way towards the gill filaments. Now, talking about the gill filaments, each gill bears two rows of gill filaments, which are called as a primary gill lamellae. So these are primary gill lamellae toward the outer side. This is the outer side. They bear the primary gill lamellae. So these two are the primary gill lamellae. In most teleos, the interbranchial septum. This is the interbranchial septum here. What is shown here, interbranchial septum, this very portion which I am making red. So this very portion, this is the space between the two you can say lamellae, that is the two primary lamellae. This is called the interbranchial septum. It is short in the case of teleost, but in the case of Labiorohita and Hilsa elisha, they are extended somewhat down, somewhat up to here, halfway down up to here. They can extend down. So these also depend upon the now, talking about the gill filaments, when we talk about the shape, size, and number of the primary gill lamellae, these are the primary gill lamellae. They vary in fishes with diverse habitats. The fishes with having the living in the diverse habitats, they have the little bit variation in the primary lamellae. The species that lead active life and depend entirely on the aquatic respiration have the larger number of long gill lamellae, are the gill filaments, as compared to the fishes which with sedentary life or having the air breathing organs. So when I talk about these very lamellae, these are very much large. These are very much large in the case of those very fishes which live in a habitat which require the active mode of living, such as hill stream fishes. In those very cases, the primary lamellae are longer, whereas they, in the case of the fishes having the sedentary mode of living, they have the shorter lamellae. But in the case of hill stream fishes, the primary gill lamellae many a times undergo division as well as division, so that they give rise to additional lamellae, so that the surface area is increased and more oxygen is exchanged, gaseous exchange takes place. Sometimes lamellae bifurcate in the middle or base to produce two or more branches, and sometimes four branches are formed. So these additional branches increase the surface area as an adaptation to the hill stream fishes. So talking about the secondary lamellae, what I told you that on the for example, this is the primary lamellae, these are the primary lamellae, and on the primary lamellae we are seeing here that these are the outgrowths. These are the outgrowths like this. This is the primary lamellae here. This is the primary lamellae. On these very lamellae, these very outgrowths are present. We call them as a secondary lamellae. So each primary lamellae bears large number of the secondary lamellae on both sides. It is not only the upper side but on the lower side as well, on the both sides. This is the primary lamellae on the lower side as well as on the upper side. The secondary lamellae are present. These flat leaf-like structures are the main seats of the exchange of the gases, and they vary in shape and size, as well as the dimension and density per unit length. They also vary, and according to the this is again according to the mode of the living, or according to the habitat. For example, here we are seeing that these are the primary lamellae, and on them these are the outgrowths. These are called as a secondary lamellae. The main function of the secondary lamellae is to increase the surface area, and herein what we are seeing here, that these are the branchial arterioles and the arteries which are flowing through these very primary lamellae, and these are actually the this is actually the flow of the blood, and in the opposite direction there is the flow of the water. So it is the counter current mechanism by which the exchange of the gases takes place. That we shall be dealing in the next part, that is the respiration in the fishes. But herein we are just having an introduction about how these very gills look like. So for example, herein we have water current has been shown to move from this to this direction, whereas blood is shown to flow from this to this direction. I will show you by the blue pen. So this is the direction of the flow of the blood, and this is the direction of the flow of the herein flow of the water. So they flow in the opposite direction, so that exchange of the gases can take place efficiently. Now, secondary lamellae are having two sheets of the epithelium, which are separated by space, and through these spaces, blood circulates. So they are having the two sheets of the epithelium. They are having the two sheets of the epithelium, The epithelial sheets are separated by a series of pillar cells. So there are certain pillar cells which separate these epithelial cells. The epithelial cells are separated by these very pillar cells. So each cell consists of the central body and is provided with extension at each end. So they are having a central body and extension toward each end. These are the epithelial cells separated by the pillar cells. Now, there are certain glands which are also associated with the gills that we shall be going through a little bit. We shall be having an idea about these very things as we are dealing with the structure in the case of structure of the gills. So these are the branchial glands, are specialized cells of the epithelium. Actually, these are the epithelial cells and are glandular in nature to perform different functions in normal and as well as the experimental conditions. Their function varies a little bit. The most common specialized branchial glands are mucous glands. So these very branchial glands are mucous glands and acidophilic granular cells, and which we call it as the chloride cells as well. So talking about the mucous glands, these are the mucous glands. These are the mucous glands. So these are the unicellular. These are the unicellular. These are pear shaped, with the neck through which they open outside. They open outside by a narrow neck, and the nucleus lies at the bottom of these cells. So at the bottom, the nucleus lies, and they are typical goblet cells. They are present throughout the epithelium. These such kinds of the mucous glands are present, and they are also present through the epithelium, that is the gill arch, gill filament, and secondary lamellae. They are present all over them. As we all know that mucus is very much important so as to prevent the bacterial infection, so as to have the smooth flow of the water as well. So these are present along the side. That is why whenever we touch the gills, they are somewhat very much sticky, having the mucus like substance on them. So the function of the mucus is the protective in nature. They protect the gills from abrasion or any other kind of the, you can say, impurities or the things which can injure the gills present in water. They protect them from the injuries, and they reduce the friction. They are anti-pathogenic. They are helpful in preventing the bacteria from or the fungi from growing on the gills. They help in the ionic exchange as well as they help in the gas and the water exchange. Now lastly, the chloride cells are also present in the gills. These they contain the granules which take acidic stain. Actually, they take acidic stain, and hence they are called acidophilic cells. They provide large number of the they are provided with the large number of the mitochondria, and they are known as ionocytes. They helped in the ionic regulation in the euryhaline and stenohaline fishes. So in those very fishes, for example, certain fishes which cannot tolerate the extreme, you can say, change in the salinity of the water, they help in those very fishes, or in those very fishes wherein they have to move from the less salted or from the fresh water to the marine water. They are also helpful. So chloride cells help in these other fishes. And lastly, the taste buds. They are the flask-shaped multicellular structure and are distributed in the epithelium of the gill arch region. And there are two distinct types of the cells. One are sensory and supporting, which help in the fish to sense the nature of the food particles contained in the water flowing through the gill during ventilation. So these very taste buds, they help the gills, they help the fish to sense the nature of the food particles which are present in the water, and they are flowing through the gills during the ventilation. They help to know about that very food particle, that what is the taste of those very food particles. Thank you very much. This was all about the structure of the gills, particularly the teleostean gills. Next time we shall be dealing with the respiration in the fishes, and particularly we shall be talking about the physiology of respiration in the case of fishes. Thank you very much.